start recording. If you want to go ahead and get to your first slide. So hi everyone, it's Tuesday night, December 18th, 2012, and this is our hands-on ideas number 5 that we're calling Printing Plastic, Candy, and Cells, and that will all make sense here in just a second. Joining us is Jordan Miller. Jordan is joining us from Pennsylvania, where he's in the Department of Bioengineering at University of Pennsylvania, but he's also very active in the makerspace community. And he is going to describe a really interesting project that I encountered online and asked him to speak to us about. We're actually playing with 3D printing in the hackerspace and playing with printing chocolate and sugar, like as in cotton candy, kind of has an interesting corollary to bioprinting in his work to be able to eventually print organs and tissues to replace those that are no longer able to be used in the human body. And so the format of this will be that Jordan is going to tell us what his background is and what inspired him and what he's done and kind of how we can think about building on his work. As a note, this entire presentation is being recorded and it'll be available for archive on our website as soon as we can put it, encode it and put it over there, as well as links that Jordan will refer to in this. So if this is your first time doing a hands-on idea session, welcome, and grab some friends and let them know that it's going on, and maybe you can build on their work. But I'd like to go ahead and introduce at this point Jordan Miller. Jordan, thanks for joining us.
All right, yeah, thanks, Dan, and thanks everyone for joining us. I'm really excited to tell you about some of our work, as you mentioned, Dan, printing plastic, candy, and cells. So I thought I'd start off by asking a very simple question, and that question is, what is the future of human medicine? And if you think back about 70 years or so, the future of human medicine looked something like this. And this is the idea of the bionic human. And we're very familiar with this from television and from movies. And this has all come out of the prosthetics industry. So for people that need, for example, a total knee replacement or a hip replacement or a pacemaker, they have now cochlear implants to enable you to hear, and people are working on retinal implants now. And so this is these types of therapies have had really dramatic improvement in the quality of lives of these patients. But if you think about it, most of these materials that are being used, they're based on metals and they're based on plastics. And what happens is these never fully integrate with the body. These materials are foreign, they're seen as foreign by the body. And if you get them when you're young, they're not going to grow with you because they're static materials. And so you'll have to get them revised or replaced over time. In addition, there are a lot of therapies that simply can't be addressed with any type of device. So instead of looking at devices, there's a new kind of idea or imagination of what the future of human medicine could look like. And it's that of regenerative medicine. And the National Institutes of Health has put forward this idea of regenerative medicine, and they've chosen as their mascot for this process actually Prometheus from Greek mythology. So Prometheus, shown here, Prometheus, he was chained to a rock and sentenced to have his liver eaten every day. This is not the most optimistic vision for the future of human medicine, but what gets the NIH excited about this idea is the idea that every day after his liver was eaten, it would naturally regenerate. So here you can see the idea. So there are all these organs here. So there's the liver, the kidneys, the pancreas, the heart, the lungs, maybe even brain tissue and muscle tissue here, that maybe we can't use devices to replace these functions. A lot of these functions of these organs are biochemical based, they're mechanical based. They have very deep integration into the biochemistry of your body. And so this idea of regenerative medicine it's really based on the idea that cells are going to outperform devices. So those of you in the maker spaces working on things, you may think, well, how are we going to make something like a device that we can implant in the body, and how are we going to make it replace some of these tissues? And really, the idea is not about making metals and plastics to make a device. It's about using cells to replace the natural function. So if you can make or design a cell-based therapy, so maybe taking a bone marrow sample from an adult human, growing up a bunch of cells, and then replacing those, trying to build organs and organ systems, then theoretically, they would grow with you. They would be part of your own body. Your body would automatically kind of accept them. And that is really this vision that a lot of people in our field here, are working on. And this idea of regenerative medicine has already had many successes in human patients. So this is actually at the top here, you see this is this very thin sheet. What's interesting about this thin sheet is it's actually seeded with cells. So they take some skin cells from someone who, for example, might have had burns over many parts of their body. They can take a small biopsy of skin areas that were not damaged. They can expand those cells. They can grow up many, many copies of those cells, and then they kind of sprinkle them over this type of thin sheet material, and they can use that to help replace damaged skin areas. There's in the lower left here, you see this corneal replacement that's being used. And on the right here, this is an artificial bladder. So you can see what looks like sewn areas here. There's actually another 2D kind of flat sheet-based material that's then sewn, basically just stitched up into this 3D shape. They can again seed these with a patient's own cells, and then they can implant them back in the body and try to help replace bladder function. But what you notice is all of these types of therapies are all based on thin sheets of material. And the question is, how are you going to make organs and tissues that are more 3D, that are more like the solid organs that you might find? And so here's another schematic from the NIH, from the National Institutes of Health. And we're not talking on the left here about delivery of DNA and genes. We're not talking about gene therapy today. We're talking about here on the right side, the cell-based delivery. So this is the area of stem cell biology. So you could think about taking embryonic stem cells. Embryonic stem cells have, of course, many ethical considerations that people are wrestling with. But now people are looking at basically adult cells, so cells directly from the skin, for example, that can be turned into something that is more embryonic-like, so they can create differentiated stem cells from there. So if we can take cells out of the adult, we can grow them in a petri dish, as shown here, we can expand them up. And that's been known for many decades. And once you expand those cells, the question is, where does the breakdown? Why can't we make, for example, this 3D liver? And the answer is it's this last step here. This final step is where the breakdown happens. So if you take a bunch of stem cells, or tens of millions of these cells, so the idea is that maybe the size of the liver, you might need something on the order of 1 billion cells. So even if you could grow up that many cells or isolate that many cells, the issue is about 95 to 99% of the cells will just die immediately after, within about a day after injection back into the person. And the reason there is that we think is that the reason is that we can't yet recreate this architecture, the very complex architecture that you see of the solid organs. So why is that such a big deal? If we can already grow or isolate billions of cells, why can't we just put them all together in 3D? And it may not seem that complicated of an idea, and the reason is because we've seen examples of this in our daily lives where we think that maybe it's not such a hard problem. So it's kind of this funny analogy here. So if you go to the deli, what you may not realize is that actually chicken doesn't naturally grow as a solid sphere of cells and tissue. And the reason is that because all the cells, if you pack them together in 3D, they can't access nutrients and oxygen, and we have no way to keep them alive. And there's another analogy that I wanted to bring up, and it's that of baking bread. So actually, oxygen delivery to, if you can imagine like a 3D sphere of packed cells, is very similar to baking a loaf of bread. So when you take a loaf of dough and you put it in the oven, you'll get a crust layer that forms. Now, chemically, this is oxygenation. This is exactly the same process that's happening in your body or that would be happening if you were to pack all these cells together in 3D. And what you see here is as you know from baking bread, if any of you have done that before, it's pretty fun. So it doesn't really matter how hot you make the oven, you're always going to get this crust layer that forms, and the crust is always going to be darker than the inside. That's because all the oxygen gets taken up by this sort of outer thin layer around here, and all the center gets much, much less oxygen. So if you imagine packing literally billions of cells together in 3D, you'd only get oxygen delivery to the outside. And many of these cells that we're interested in, like the liver cells, for example, they require enormous amounts of oxygen. And the challenge is really how are we going to deliver oxygen and nutrients to the center of these types of constructs that we're making in the laboratory. And so if you really want to look at the architecture of these solid organs, what you find is that these solid organs are not actually solid at all. So the solid organs, for example, the human liver, it's a very large organ, but almost half of the volume of the liver is blood vessels and blood vasculature. And this vasculature has this very complicated, interconnected, multi-scale structure. So you see on the right here, this is micro CT, so this is a kind of a CAT scan done of a liver tissue. And you can see this very complicated architecture. And the reality is that we don't know how to make structures like this. So the body is very good at making structures like this. And this happens very easily during development, during embryonic growth. But we don't know how to make stuff like this in the lab. And those of you in the maker spaces, in hacker spaces all around the world, actually, you may think, well, why don't we just 3D print that type of structure? And that's actually a big area of research going on right now. So that field is called bioprinting. And you see an example of bioprinting here. So you can imagine 3D printing, as you may know, is where you're, instead of 2D printing, which you think of maybe ink on paper, you're printing one layer at a time, and you're reproducibly putting one layer on top of another to stack it up in 3D. And this is how some people are looking at trying to build tissues and organs. So what you see here is on the left side, you see this is actually extruding through that metal tube. They're forcing cells and gel. This gel is kind of, mechanically, it's kind of like jello. It's a sort of support material that is providing structure. So the idea here is you'd print or you'd extrude cells and gel. You do it one layer at a time. You kind of build up these structures, which you see more on the right. After a couple dozen layers, you can see these kind of structures being formed. And again, there's a lot of people working in this area. But from our perspective, we were really interested in many of the interesting cell types for human therapies, so cells like liver cells. And it turns out that while liver cells are very interesting and important from a potential for a therapeutic perspective, something we might want to put back into a human patient someday, the liver cells actually inherently, they're very fragile cells, and they don't handle extrusion well, and we don't really know how to change the cells to be able to make them survive this process fully. And
Jordan,
so,
it's Dan. I could ask a
oh
quick
yeah,
question. Can you give us an idea of the
sure.
scale that we're looking at in this image?
Oh, sure. Yeah, so the nozzle here, the tip of the nozzle, the diameter, the inner diameter of that nozzle is maybe around 1 millimeter.
Okay.
This is the publication here to the bottom right if anyone wants to read more about this publication. We didn't do this work, but this is work that we're familiar with.
So
Um,
this is like roughly a quarter of an inch in diameter,
That's
this
right.
image.
And you may only have thousands to a few million cells in a structure like this. So you can get some cell types to survive this process. You can get cancer cells to survive this process. You can get skin cells to survive a lot of this type of process. But a lot of those cell types, like liver cells, they just won't survive this process. They really are demanding oxygen and nutrients all the time.
Okay.
So we
Sure.
kind of looked at this. Oh yeah, go ahead.
I said, no, just thanks.
Okay, great. Yeah, so we kind of looked at this this sort of issue in the field, and we thought, maybe there's a different way. So this is where we first kind of approached this idea or this problem that is happening in biology and biomedical research labs. And we didn't know really the best way to approach this. but we were kind of looking for a different way. And one thing that I noticed, so I ended up going to the Body Worlds exhibit. So Body Worlds, if people have seen it, Body Worlds is really an art exhibit where people have sort of willed their bodies after they die to this artist, and what he does is he will process the cadaver into an art exhibit. And this is one thing that is on display there. This is whole organ vasculature. So what happens is they take, for example, a human kidney, like shown here. They can inject in the blood vessels this kind of silicone that will harden and cross-link. It's kind of like an epoxy that will harden in all of the blood vessel networks, and then they can dissolve away all the rest of the tissue. And you can see these types of amazing architectures that are just on display there. And so when I saw this, I had this epiphany of like, really, this is the architecture. This is really what we want to make from scratch. So what if we could make this first? If this is really the most important thing, if this is going to be the highway for nutrients and oxygen to be delivered to our cells, maybe we can focus on making this first. And if we can pick the right material, maybe we could think about putting the cells around there and then sort of do the inverse process, sort of cross-link the cells in a gel around this type of structure and then dissolve it away afterwards. And so that idea was kind of, it's kind of a weird idea, but it was kind of kicking around in my head. And I had been to a local hackerspace here in Philadelphia, Hive 76, and you see on this upper right panel here, this is what a 3D print looks like coming off of these commercially available and open source 3D printers. So one of them is called a RepRap. That's one of the ones that we're really interested in for this project. But what you see is, you know, it's not that similar to the architecture here, but it maybe is somewhat reminiscent of the types of structures that you want to make. You have these cylinders here, they're all packed together, kind of layered on top of each other. You could maybe think about using something like this to build up a structure like this. But if you think about it also, these types of materials are not really ideal for use here. So this is this epoxy resin I mentioned on the left side. And these 3D prints are these bioplastics that are coming out now. You can use ABS, which is essentially Lego plastic, but you can also use these bioplastics. And the bioplastics are biocompatible from the sense that cells can grow on them. But anything you'd use to dissolve these plastics, they're very caustic materials. And even anything to dissolve this epoxy resin, they're also very caustic, and they would just kill your cells immediately. So again, we have this idea that if we were to put liver cells around these types of structures and then try to dissolve them away, we'd probably just kill the cells also. So that was kind of where we were. And then about a year later, I had dessert. And what you notice here, you should always have dessert. So don't look at this flan here, but if you look at this sugar cage here, there's a sugar cage that the chef has made kind of draped over. It's self-supporting, it's freestanding. And again, this is maybe somewhat reminiscent of this type of upper right 3D printed structure from a commercial or open source 3D printer. So this is kind of the idea. So if you think about this sugar material, we know it's already biocompatible. It's already in your bloodstream right now. It's extrudable. So maybe we could modify a 3D printer to extrude this type of material, maybe make a shape something like this open lattice here that's really well defined, very reproducible. We can have the machine do it for us, and then maybe we could use that for something somewhat equivalent or analogous to natural human vasculature. So that was really the idea that we decided to start on, and this is about 2 years ago at this point. So how did we do this? So we used actually, we have many collaborators, many people working on this, both at University of Pennsylvania and our collaborators at MIT. But I want to emphasize this is really a global project, and we utilized many open source cross-platform toolkits, which you see listed on the right here. And each of these projects, they're not really necessarily optimized to work together, but because each of their communities they believe in open source. They put forward all their code. They have many helpful tutorials online for how to use them. We were able to modify a lot of these, taking advantage and getting some feedback from the community on using all of these tools together to be able to extrude and modify them to extrude sugar for these types of biocompatibility studies. You can see here in the bottom left, this is our Hackerspace Hive 76 in Philadelphia. And this is where a lot of the fundamental work was going on for this scientific project, actually. So it was really great, the ability to just go into a local makerspace and say, hey, I don't know anything about electronics, but this is what I want to do. I want to be able to modify this RepRap open source 3D printer to be able to extrude sugar. And everyone there was really excited to help out. So we have kind of this schematic overview to kind of, again, I just want to emphasize exactly what our plan is for kind of reconstructing things that may function like natural human tissues. So we're starting here at the upper left with this kind of 3D printed filament network, and we're going to try to make this out of sugar. And if we pick that sugar material just right, not only will it be biocompatible, but it'll be strong enough to support itself in this kind of open lattice structure that I've kind of schematically drawn in the upper left. So then after we draw this, or we kind of print this, 3D print this freestanding sugar lattice, we're going to then pour cells and gel all around that and encapsulate that sugar lattice. Now the gel is going to solidify and the solid sugars are then going to liquefy, and then we can flow all the sugars out of that architecture. And then everywhere there was a sugar filament, that becomes this perfusible, essentially blood vessel analog. So it's cylindrical, just like your blood vessels. We can make it many different diameters. And this is really the idea, that this sugar material is going to be both a structural analog, but also be biocompatible to be able to be dissolved away without killing cells. We can just put it in media, we can just put it in basically saline, so any kind of salt water, and all the sugar will dissolve and we can flow it out. So this is really the idea that we started working with. So I had mentioned the RepRap 3D printer. So when we started, the RepRap 3D printer project was pretty new. It's a low-cost 3D printer. You can legally and freely download all the software and hardware designs for this open source 3D printer. It's really quite remarkable. Anyone can contribute to the project. And there's this issue, I guess, is very similar to the Linux business model for the Linux open source operating system, which you may have heard the internet meme that Linux is free if your time is worthless. So to some extent that was somewhat representative of what it was like in the very beginning. We were kind of on the bleeding edge there of how to figure out how all these parts fit together to get these open source printers to work. But I think this is a very mature community now, and it's quite easy for basically anyone to get started with RepRap now. There's there's probably 100 different kinds of tutorials online. There's dozens of different types of 3D printers that are all considered part of this RepRap 3D printer family. And when we got started, we had just done a couple contributions to this RepRap community, this open source community. So we had this program shown on the left here. This is called SkyneFox. So this is something that I was writing to help kind of keep all these configuration files in this Git repository to be able to manage, basically file management for a lot of the different settings you might want to use for 3D printing. And then on the right here, this is the first heated build platform that was being used. What people don't know about the heated build platform that is found on these 3D printers nowadays is it was actually designed to be able to print sugar for this project. So we did the first design, and part of the issue with sugar is it's what's called hygroscopic. And hygroscopic things, they can pull moisture from the atmosphere, and they'll just, if you imagine that open lattice structure, that will just kind of melt just by being left out in ambient conditions. So instead, we wanted to use this heated build area to keep it dry and keep it warm. So once we had these types of tools in place, if you want to program this type of code, it's very simple. We're using this open standard called GCode. And GCode is basically just an ASCII-based text file. And you can see an example of that here on the right. So it's just a series of lines. Each line here is one command. So here it's saying, grab tool 1, go to this XYZ position at that speed, and then open this valve, and then go to this position at that speed. And this is how you build up the instructions that the sugar printer is using for building up these types of structures. So you can see an example of that here. There's this movie here of this is our sugar printer in the lab at University of Pennsylvania now. So we're doing this for these sterile, basically use for culturing cells. You can see the sugar filament here coming out here and being supported by other sugar filaments, and they're kind of melted together and they get fused. This is me injecting a gel all around that sugar vasculature to be able to be used for these biocompatibility studies. So I just want to highlight some of the work that we did with this type of printer. So we can print many different diameters of channels. So just like the native vasculature in your body, there's many different sizes of vessels, and they're all connected together. So one of the things that we wanted to do is be able to print many different sizes of vessels. And the way that we're doing that, it just follows this very simple power law that's derived from glass fiber drawing. So if you know optical fibers for fiber optic cable, this is how they're made. This is the same way. So you can heat it up and you can pull almost any diameter of filament that you want. So we can just change the speed that the extrusion nozzle is traveling, and that allows us to print all of these different sizes of vessels. And you can make these really interesting structures. So just like those 3D printed bioplastic prints that I showed in the beginning, you can do all those same types of structures with this sugar-based glass. So mechanically, it is a glass, and that has some really interesting components. So it's optically clear, it diffracts light in these very interesting patterns, and you can connect all these different sizes of vessels all together. You can see here on the bottom left, just like you might fuse two glass rods, so if anyone's seen glass sculpture being done live, you can also fuse these sugar glass filaments, and they will fuse just by surface tension. So this is just really interesting, very smooth fusions here, and this is going to allow us to make these connections between neighboring vessels. So here is kind of just emphasizing that at these intersections between the filaments, so these interfilament fusions, when we pour this gel all around those sugar filaments, the gel will solidify again, the sugar will dissolve away. Everywhere, all this architecture becomes a perfusible network. So when two cylinders overlap, you get an oval. And this oval becomes this intervessel junction that's fluidically connecting all of the vessels together.
Jordan,
So
this
to
is
build
really
up
interesting.
tissues,
What is the print speed on doing this? Is this fairly? I mean, we saw the video, but is this fairly complex as far as a print build? You know, when you think about 3D printing, or is this looks like it works fairly quickly?
yeah, it's actually about the same speed as any kind of 3D print you might do on your own with these bioplastics
So,
and with the Lego plastic. So it's on the order of minutes to print one of these structures.
so the
The
so
sugar
the
print, it's all, oh, say again?
so the intricacy that we're seeing here of these interconnections doesn't really add to the print time. It's just like you were printing a solid.
That's right. So everything that we're doing, it's all based on essentially the temperature gradient. So how hot is the nozzle and how cold is the ambient temperature? And if we're blowing a fan on the sugar itself as it's being extruded. So it's going from this molten liquid state, and we're literally freezing it in place. It's weird to think about freezing it at room temperature, but you have to get it to around 110 degrees Celsius to be able to extrude it. So when you cool it down rapidly to room temperature, it'll just immediately freeze in place. And it's strong enough, and we kind of reinforce this sugar glass to be able to stand up on its own.
Great, thanks.
So yeah, it's been really a fun project. So then, similar to bioprinting, where we might want to make essentially different zones inside of a tissue, we can do something similar to that in this type of setup here. So what you're looking at here on the bottom, so at the top here, this is just showing that we can sort of mold these gels into any kind of material. So they have all different cross-linking mechanisms, but they all work very well with this type of sugar printing. And then in the bottom here, this is a cross-section through one of these gels where we've created zones. So what you're looking at here in the blue, these are these cylinders, these are these perfusible vessels that we're making out of the sugar. They're molded after the sugar. Every green dot you see here, every tiny little green dot, that's one single cell. And what we're showing here is that this 3D space all around this vasculature, we can make very complex essentially gradients. So different concentrations locally of cells. We go from a low cell concentration to a high cell concentration. you can imagine maybe you go from one cell type to another cell type in this kind of smooth gradient. And that can all be done independent of this blood vessel architecture that we're making. So this is just kind of showing some of the flexibility of the system that we're doing. I think one thing I was really excited about, this is a movie. You can download this movie from that link that's posted on Openly Disruptive. This is a movie of pumping human blood through these vessel networks. So not only are they fluidically interconnected, we have that sort of oval connection here between these two vessels, but the gel that's cross-linked all around these vessels now, it's a very strong gel mechanically, and the gel itself can support positive pressure and pulsatile flow, just like what you'd see in the human body. And then similarly, so that was just for one junction. What if we look at a whole network? So we have this kind of lattice or ladder architecture here, and you can see that this blood is being flown in here in the upper left, and it goes through these interconnections here down to this larger tube at the bottom here. This is again under very high pressure, the same pressures that you might find inside the body. So another thing that we wanted to do with these types of architectures, so there are cells that line all of your blood vessels, and they keep your blood from clotting, and they also regulate sort of the nutrient and oxygen transport. So you can imagine if this is oxygen coming through these lattice networks, then that's going to just sort of start diffusing or permeating through the wall here of this gel and kind of nourishing cells in the bulk. But in your body, there are also cells that are there that regulate that, they control that, and they also are responsible for keeping the blood from clotting. So what we did is we took some of those cells and we labeled them in red, and we injected them into all the blood vessel networks. So you can see that here in red, this sort of crisscross structure. This is that lattice architecture, that those vessels, and so they have all these red cells lining all of them. So the scale bar here, this distance here, the diameter of these large vessels is around 500 micron. So 500 micron is half a millimeter. You see the scale bar here. The scale bar is 1 millimeter. So you can get an idea of the scale here that we're talking about. And so we're lining all of these, and all these green cells, again, so every dot here in green, this is another one of these living cells that we're suspending in a gel all around these vessels. And so this is just immediately afterwards, after we've made these gels, we've injected these cells into the vessels. And what you see here is that those red cells, they line all of the vessel networks just like you might see in the body. So not only can we line these large vessels and these small vessels, but we can line across this, this intervessel junction, this fluidic connection between these two different vessels. And this is all empowered by this rapid casting from this sugar-based architecture that we made. So I have a movie here of what this cross-section looks like. So this is similar to CT, so sort of a CAT scan. This is essentially an optical CAT scan. We're taking optical slices through this tissue. So you're kind of looking in cross-section kind of deeper and deeper through this tissue. There's a very thin optical section, so you can kind of see there's the inside of that large vessel. This is where it connects to that smaller vessel, and that smaller vessel is at a higher position in the gel. So that's how you see it there. And so we had this debate, you know, how much of the blood vessels do we need to make to be able to keep cells alive? So in your body, there are vessels several millimeters in diameter all the way down to about 10 microns. So that's the size of capillaries. So do we need to make a capillary network? We didn't know initially. So what we did is we kept growing these types of tissues that we made. These are living, essentially living tissues with living cells in them. And we wanted to see what would happen with these blood vessel cells over time. And so if we take this smaller vessel, now we're going to kind of rotate it 90 degrees into the plane of the screen, and we're going to kind of look down one of these channels, another one of these optical sections. And this will be after several more days of growing. And what you see here is these red cells are lining, but they're also creating these new capillary sprouts you see here, all these kind of red sprouts coming off of these vessels. They're growing here, essentially new capillary sprouts coming off of the vessels that we made that were patterned off of the sugar that we 3D printed on a RepRap. And you can see that in more detail here. So again, we have this, this is the in cross-section, this is the hollow cylinder where we have all these red cells lining them, and they're able to create their own, essentially, the beginnings of capillary sprouts. So we think this might be a way to get these very complex networks where we can pattern vessels down to the size here about 200 microns, so 2/10 of a millimeter, and then let the cells create their own capillary network off of that would then be fluidically connected to everything to keep all these green cells that are all around these vessels alive. And you can see also, it looks like these green cells are starting to kind of grow and migrate towards these vessels. We haven't looked, we're going to look at this in more detail, but it just kind of, it looks like that might be what's happening. So that was pretty exciting. So now, how are we going to use this to deliver nutrients and oxygen to the cells? How are we going to measure that? So if you remember back when we talked about baking a loaf of bread, we can do the same type of assay, the same type of study, essentially as baking a loaf of bread, where we're looking for basically cell activities. You can imagine the crust of bread as that's basically where the activity, where the oxygenation is happening. And what you see is if you make a very large gel, like those gels we were talking about, making a very, so we have a thick slab of gel here. So this is 20 millimeters across, and going from left to right. It's about 5 millimeters in up and down, shown here. And this is just a cross section. And what you're looking at is this way of imaging the cell activity. So again, every tiny dot that you see here is one cell, and the brightness that it has on the scale is representative of how active it is, really, how much protein is it making. So there are cells here everywhere in the center, just like there are on the outside, but you can see by their color, they're really not active at all. So if we're trying to grow these tissues, these thick slabs of tissue, these kind of solid organs, you can see that we can't just keep making it bigger and bigger. Eventually, if we go above a couple millimeters in depth, then we're going to start to get a lot of cell death in the center. So in contrast, if we look at a gel where, again, in cross-section, where we have our perfusible channels, these are these channels, these fluidic channels, these blood vessel analogs we made by molding around this sugar architecture that we 3D printed, then we can flow nutrients and oxygen through those channels. And you can see here essentially rescue or sustainment of the activity of cells all around these vessels, and it directly matches the vessel architecture that we made. So this is sort of one example of how we might be able to use this type of architecture to be able to deliver nutrients and oxygen to living cells in these much larger scale and much higher density than higher cell density than what other people have been doing before. So I just want to highlight in more detail what those vessels look like. So again, so every single dot that you see here is a single cell. And again, I made these images, I processed these microscopy images with some more open source tools that are shown here at the bottom. So there's hugin.sourceforge.net and ImageJ. And we're just going to kind of pan across here. So Hugin is actually a panorama software people use for their vacation photos to stitch them together. It actually works extremely well for stitching together microscopy images. So I take about 100 microscopy images of these gels, and we can stitch them all together in this seamless panorama view. So you can see all the activity of all the cells. So again, at the outside of the gel, they're getting nutrients and oxygen just from the atmosphere. But deeper in this gel, so again, this is in cross-section, what you see is one of these channels where we're pumping nutrients and oxygen through there, then coming out into this bulk gel, you can see the survival and this activity of these cells deeper within this tissue. So we were really excited about this. And then you can think about what would be maybe the next step. So how are we going to get these more complicated blood vessel networks? And this is one thing that we're working on right now. So you can imagine if we take that whole organ vasculature that we showed from before, this sort of CAT scanning, sorry, this blue rectangle is off center, but if you blow up one of these structures, you can see that we can convert this type of micro CT, this type of CAT scan, into a 3D printable file. And that's shown here in the bottom left. So the problem here is that this is kind of a very complex scanning algorithm, and it's very hard to make something that is processed well. So one thing that we're looking at is maybe we can just do this to analyze what the vessel architecture, what the kind of idealized vessel architecture might be. And then if we do that architectural analysis, we can then regenerate a tree-based structure from scratch on the computer, and maybe then it would have very nice sort of topology, something that you might need for 3D printing. And those people that have done 3D printing before will be familiar with this. You want to have this sort of manifold structure that you've designed on the computer from scratch, and that will translate very easily into 3D printing. And then, instead of 3D printing in plastic, we're thinking about eventually printing this in something like sugar, one of these biocompatible sacrificial materials. So sacrificial, like we're just going to melt it away or dissolve it away afterwards, after we put cells all around there. So I mentioned also that a lot of this work, we started off by doing it in our local hackerspace. Well, this extruder that extrudes sugar, it also extrudes other things that are edible. And one example here, this is in our hackerspace. This is extruding solid tempered chocolate. So this is just something that was fun that we started in the hackerspace too. So we're kind of looking at ways to maybe adapt the same type of technology for things like just making some holiday treats or something like these edible foods, and using this as a local hackerspace project. So if anyone's interested in doing this, all the designs that we modified for the RepRap to be able to extrude sugar and chocolate, we put them back online, and there's some links at the end you can find that will show you, kind of lead the way on how to do that. And if you have any questions, you can contact me. So in addition to the sugar, there's in addition to the chocolate, we can also do sugar. So this is some examples of one of those same types of 3D printed files you might find on a website like Thingiverse. So instead of printing it in plastics, here this is an octopus that we're printing in our hackerspace. This is printed completely out of sugar. And you can see that, again, this sugar, it has this glassy architecture. It becomes this very clear, amazing structure to look at, and you can think about flavoring it differently. So that's been fun. And if anyone's heard of Cory Doctorow from boingboing.net, so he actually recently, he visited MakerBot, which is a company in New York who's commercializing some of these open source 3D printers. They have a 3D scanner. So they have, so similar to that micro CT, it's a lot more crude. They're just using basically photographs to map your face, and they can make that into a 3D printable file. So Cory, he had his head scanned. He put his head file on Thingiverse, so we downloaded it, and you can see here on the left, this is it printed in plastic, and here we're printing it in the sugar architecture. And he actually came to Philadelphia, and we presented him with it. So this is just him tweeting that now he's candy. So you can see about the scale there. So this is about tens of or a couple centimeters across. So I just want to end just with the idea that the reason that we're doing this is really about human health. But I think what's really interesting from a sort of public perspective is also the way that we're doing that is we're reaching out to the open source community, and they've been extremely helpful helping us get this work done. And all of the contributions and modifications that we've made to this project, we put back online. So it's really, we're hoping it'll become more of an open standard for the sciences as much as it is an open standard for kind of hobbyist 3D printers. And so, the challenge here for human medicine is that despite really thousands of years of investigation, the tissues and organs in the body are still unbelievably complex. And I wish I could say, exactly what we need to do to be able to get these structures, these very complex architectures, be able to make them. We don't know which components here. So if we look at the organ systems, we can kind of expand that. We can look at the tissue level. These very complex architectures are present in most of these solid organs. So here we're looking at a blow-up schematic of the liver. We can look at these subunits here of these very complex canals and fluidic channels and cell arrangements. And we don't know all of the different architectures that might be needed to be able to do this. We're approaching it from an engineering perspective. So we're trying to say, can we start from scratch? What are the minimum set of features we'll need to build back in to a structure like this? And how are we going to do that? So if anyone's interested in this type of research, I think that it's very ripe for investigation. There's a lot of people working on this type of area all around the world, and certainly if people have any ideas, I'd love to hear them for pushing this forward. And I think you know, you don't always get the best inspiration from just being at work, and I think it's really a success story for you know, both art and the open source community, that a lot of the inspiration for this sugar-based printing came from outside of the lab. And I think it's just another good example of the way that kind of disparate technologies and disparate kind of disciplines can all feed back to help progress and kind of inform other fields. So with that, you know, I'll be happy to take any questions. I have a bunch of links here if people want to see more. So the top link here, this is our lab at University of Pennsylvania, reprap.org. This is the 3D printer that we're using, so you can find all the design files on there that we're using and that people have used in the past. And there's tons of different designs out there if you're looking to do this. Hive76.org, this is our hackerspace here in Philadelphia, where we did a lot of the initial work and a lot of the refinement of the sugar extruding process, and now we can do it with both sugar and chocolate. So that's kind of fun to do from a hackerspace perspective.
You have to move on to caramel and then and toffee next. You know.
That's right. Yeah, yeah. I think there's a lot of things that would be interesting to do. Thingiverse here. Thingiverse is a great site for. So there's a lot of people that have made different designs, so 3D printable designs, and you can legally and freely download those designs. They have different terms for their licenses there for downloading and printing. So that's where we got things like the octopus. That's where we got Cory Doctorow's head. And you can find a lot of the modifications that I've made to the 3D printer here at my my page on Thingiverse, my page on GitHub, where we're hosting a lot of the files there. And if anyone wants to continue the conversation or has any questions too, I'm available also by Twitter, shown here. So so thank you. I'll be happy to take any questions.
Great. Well, thank you very much. This was great. I have a few questions that have come in. One question early on, somebody asked bioplastics. You referenced bioplastics or biocompatible plastics, and just somebody wanted to know, are those something that the average hobbyist has access to, or is that a real specialty item?
Oh yeah, so about bioplastics. So the issue, I think, is from a regulatory perspective. It can actually be a very complicated answer. So let me just say that most of the very inexpensive available bioplastics. So one of them is called PLA. It's polylactic acid. That is, it's my favorite plastic to print with. It has its base material is one of the materials that people are using for implants nowadays. It is the base structure of it is a biodegradable plastic, and it's actually biodegradable in the body as well. So that's one of the definitions of a biocompatible material or biodegradable material. It can naturally degrade in the body into harmless byproducts. So polylactic acid is a polymer. It's actually many lactic acid molecules linked together. And lactic acid, as you may know, comes from when you're exercising, when your body is naturally kind of low on oxygen, it'll kind of go through a different metabolic pathway and make this lactic acid. So from a theoretical perspective, yeah, polylactic acid, PLA, the same plastics people are printing, are some of the same materials you may think about would be biocompatible for implantation. The challenge is that most of the suppliers of those types of materials, they have the actual exact formulation as a trade secret. So you will never know exactly what additives they have in there, and if those additives that they use to make their extrusion process easier would be compatible in the body. But what I would say is that it's a great place to start. So that's exactly how we started. We started with those bioplastics, with these PLA commercially available things anyone can buy. I think it's about $100 now for 5 pounds of this plastic, which will get you quite a long ways in 3D printing. And we started everything there. We learned how to use a 3D printer. We learned how to modify it based on the need to print plastics. And then all we did is we just changed the extruder. We changed the extruder out for this new kind of it's air pressure driven extrusion to extrude this kind of molten sugar out. But all the designs for that are online as well. So I think it's a great place to start. And there's also plenty of people making 3D printed plastic structures like those lattice structures, and they're putting cells on top of them and growing them in that type of architecture. So,
Yeah, I was gonna.
so
That's
yeah, I
a
hope that answers the question.
yeah. And another question that came up is, you know, any thoughts about how to make this, you know, get more complexity? So obviously, you know, and you pointed out, you know, that you know, an organ tissue is far more than just the, you know, the liver is way more than just the liver cells and the blood vessels. But you
Yeah.
know, some of the you know the structural elements and some of those things. Any thought that you know eventually being able to kind of create multiple layers of this kinds of structure that are kind of intertwined to get that level of complexity, or any thoughts about how to get to that?
Yeah, I think what's really exciting from my perspective for this type of technology is that the assumption is there that we will need to get that level of complexity. But I think that we don't know that for sure. We haven't yet proven exactly what complexity we need to rebuild back in to a solid 3D structure. So for example, before we started this project, everybody told me, you know, we were really going to have to make the capillary network as well. But we
Mm-hmm.
found out that the cells were able to make the capillaries and these capillary sprouts on their own. So maybe we wouldn't need to make all the way down to a capillary level. In addition, for the liver cells in particular, that's another good example because some people have done implantation studies with liver cells where they can inject them, and they're trying to find a good site for injection because you know most of the time when you inject cells, as I mentioned in the beginning, they'll die within the first day. So they were injecting liver cells into lymph nodes, and what happened is the cells were able to kind of reorient, figure out where the blood vessels were, and they were able to start secreting the natural things that liver cells make and produce. So I think that it's not quite clear, at least in my mind. You know, I can't speak for the entire field, but in my mind, it's not quite clear what level of complexity will absolutely be needed. And what I like about this type of technology and taking the engineering approach is we can sort of very slowly, very reproducibly start dialing in complexity one step at a time, quantifying, figuring out, measuring exactly what level of basically you know, provisions these cells are secreting, and then kind of see if more complexity will help. I think what a lot of people find, especially engineers, is a lot of times you can get to overly complicated setups quite quickly, and it's much harder to get to a more simple design. So I'm really excited at creating architectures and that will allow cells to kind of reorganize, and a little bit of kind of that zonal patterning that I mentioned a bit earlier, I think is possible as well.
Do you what this is a question for me. What do you see as you know is this kind of a culture clash? You know, do you see you know when dealing with the you know the folks that are you know purely on the medical side, you know, to think about actually creating these structure, you know, to creating these tissues and everything, and how the printing works and everything. Is this something that is um, you know that takes some work to get that interdisciplinary you know um, collaboration, or is this something that you know that they respond really quickly to, you know?
Oh yeah, I think what's happening right now in a lot of the medical fields, especially, is many of the sciences and engineering fields are really coming together. And excuse me, I think what you're finding is that a lot of these fields, these half of the challenge, excuse me, is just getting people to talk to each other and to get to where they can understand the language and the jargon that they're using. But I think that what we found is people immediately can see what their contribution could be. They may not see the entire picture. So a lot of the people that I worked with in the lab, we didn't know, myself included, we didn't know all the electronics that we needed to learn to be able to do this. But in our hackerspace, the opposite was true. You know, they knew exactly what things we needed to modify on the electronics, so they may not have known all of the biology that we've been studying to understand exactly what we wanted to build, but they could help us build it if we could help kind of orchestrate that. So I think it was just actually really a natural fit for kind of very good complementarity of skill sets. And I think, I guess you kind of mentioned the culture clash. We haven't experienced that at all. I think everybody in the lab was really excited that we had people to help us do the electronics, and the people helping us with electronics at our hackerspace, they were really excited to help out with the science project, which is not something we usually have going on in our hackerspace. So yeah, it was just an amazing project to be part of.
Do you see the evolution of a bio 3D modeling language or standard? There's certainly there's some uh, some spatial information and everything, but do you see combining some of the life science with that, and to some metadata, or almost like a way to start building something, a CAD for doing biostructures?
Yeah, absolutely. So one of the things that is very interesting, so in addition to the actual architecture of something like an organ or something like the vessel architecture, one of the things you have to think about and you have to calculate, there are ways to do this now, is to calculate where the oxygen delivery will be coming out of these architectures. So you want not just a CAD/CAM modeling package, but you also need to be able to understand something of the biology to understand exactly what the architecture, how that's going to impact oxygen delivery to the cells in the bulk. And there is some work being done in that area. We're working on it a bit. We've been talking to some of the commercial 3D companies about some of this stuff, and I think you'll see more of that coming down the pipe. But I think the challenge is also, trying to come up with an open standard when, again, we don't know. So open standards, I think, are best when you already know all the constraints that you have. And I think
Right.
the fact that we don't know all the things we're going to need to think about to build organs is going to make it really hard to create a standard for CAD/CAM for organ design that's probably right the first time. So I think you know, people that are working on this should really think about modularity and refinement, the ability to refine those designs over time. That will help a lot.
Yeah, you can imagine, you don't know just how much metabolic data you're going to need to carry with, every point in space. And so yeah, it would have to be somewhat flexible, I would think.
That's
Until,
right. Yeah.
well, great. Um, Last call on questions here. We're about to wrap up. Another one that I got texted to me is talk a little bit about, you earlier showed the picture, the micro, or the micro CT of a vascular network, and then showed kind of an optimized one for printing.
Hmm.
How, talk a little bit about how um, you see that whole optimization to make things that are functionally equivalent, but printable. Is that something that's a fairly smooth, a fairly easy thing to do, or do you think that that's an area of a lot of opportunity um, for for addressing?
Yeah, I think what's really interesting, also from our perspective, is that there's been tons of studies, I think over the last 100 years, mapping exactly the anatomy of the vasculature, trying to design more recently in the last 50 years or so, trying to design or understand what are those delivery mechanisms coming out of the vasculature into the bulk of the gel, into the bulk of the tissue that's nourishing cells in the solid organs, but that's also keeping things like cancer cells alive in solid tumors that also have very complicated vasculature. And I think that we just don't know what will be the best way forward for that. But what gets me excited about this type of technology is that it's extremely reproducible because the robot is making all of the architectures. So from an empirical perspective, from the perspective of actually doing an experiment, it's going to be probably one of those things we're just going to have to do kind of brute force optimization where we're going to try many, many, many different variables. But the fact is that we'll be able to change one variable at a time in a very scientific way and really prove which are the key factors that matter and which don't. So for example, we have this debate too. Are we going to need to make a direct vascular architecture that exactly matches that of the liver? Or will the liver cells be able to accommodate um, types of 3D architectures of the vasculature that might be easier to print, like you mentioned? So maybe those will be more scalable from a manufacturing perspective, and maybe like the liver cells implanted in the lymph nodes, maybe they'll just reorient and they'll be fine. We just don't know, but with this type of technology, it is now scalable enough, even in a research lab scale, to be able to ask those questions and be able to get an answer within within a year or so.
What do you think is the horizon for people to, so, oh well, I have an uncle that needs a new liver. How long till I can tell him he can get his printed at the lab? I mean, are we talking 10 years, 20 years? There's a lot of work to be done here, right?
Yeah, I would say, I would say we're at least probably about 30 years away, with the caveat that, about 30 years ago, we also thought we were about 30 years away.
Hmm.
So that's sort of the difficulty with science, is that the more we look at the biology, the more we've now realized that we don't know. So there's still so many unknown unknowns out there. We don't even know all the things that we don't know to be able to address. So with this type of system, we are starting to now be able to create structures large enough to be able to test quantitatively, be able to prove which architecture designs will be better. But the way that these things work is usually you need to do a bit of work just in the lab, what they call in vitro, so just in the lab setting, having things hooked up to pumps and such. Then you probably will need to do something like in an animal model, something that will be able to show that you'll be able to do this inside of a living organism. And then eventually you might get to something towards a clinical trial. Clinical trial would be inside of a human patient. Um, so those are usually, there's still more research and development needs to be done. Certainly a lot more on the in vitro side, let alone the animal models. But I can say that in this work, this paper that we just got published, and anyone can contact me if they want a copy of that paper. I can email it to people. Um, we are basically at the scale of tens of millions of liver cells, and that was good, and we showed that they survived a couple weeks. But obviously you want to be at the scale of closer to 1 billion liver cells, and you want them to survive for years. So that's sort of the scale that we're at. But the tens of millions of liver cells, that was already about 100 times larger scale than what a lot of other people have done with liver cells. So we were quite excited with that advance, although we still have quite a long way to go.
Great. Well, thank you very much, Jordan. Just to wrap up, also, somebody sent me a note to remind me that, and Jordan and I had talked about this earlier, that if you're in the St. Louis area or in the Midwest, like I am, uh, Modern Meadows is a company that's actually attempting to do some of the similar things that Jordan has been talking about, but with the idea of actually producing meat for market, which is, so there's a lot of different ways to think about how these technologies might work and why they might work. But their dream is of the steak that doesn't actually require the killing of an animal to have. I'm not sure how practical that is or not, but that's kind of interesting. So this wraps up this hands-on ideas. We come back in January. We have Sean Brady from Nourish the Planet out in Fort Collins, who is going to talk about a permaculture system that he's done that fits on a porch to let you have vegetables and protein for a family that fits really on the size of the typical porch. Following that, we've got some ones coming up with some people doing some really interesting urban art, including somebody who's taking over billboards in Los Angeles and turning them into aerial bamboo gardens, and a couple people working with facial recognition software on some interesting hacks. So some fun stuff, and join us and for another session just like we had with Jordan. But Jordan, thank you very much, and hope you have great holidays and you stay warm.
Thanks. Thank you so much. You too.
Take care. Bye bye. Jordan, are you still here?
Yeah.
So I muted everybody else. Do you feel comfortable doing a quick talk through, like we said, about the kind of the soundbite of what inspired you, what you did, and and where you think people could go next with this, or do you want to pick that up and do that quick sometime later this week when you have a chance to think through it?
Yeah, maybe later this week because wasn't sure exactly the scope of that, so we might want to refine it a bit.
Yeah, and I might, what I'll do is I'll send you an email with just kind of, it won't be factually accurate, but it might be a good way